All right, this is Dr. Mead, and this is the review of the cellular transport, including the cell membrane, cell signaling, mitosis, control of the cell cycle, and cancer. I'm going to take you through a bunch of sample questions to help you review. Again, this is from the biologyjunction.com website that I've recommended before. I strongly suggest the technique that I've told you about where you cover up the answers, put your hand or a card over the answers A through E, and just read the question and think about the question before you consider the answers. So I'd encourage you to take a card right now and hold it up to the screen so that you can block out the answers to the questions as we go so that you can think this process through. Let's start with the cell membrane. Here's a really easy one. I would definitely recommend that you draw yourself a picture of the membrane, number the different parts of the cell membrane, and quiz yourself on all the parts and be prepared to identify the way they look. So this question, all of the following molecules are part of the cell membrane except, and we all hate all of the above except kinds of questions, but I would cover this up and make a list of all the things that I know are in the cell membrane. Phospholipids, proteins, the cholesterol, etc. Maybe I would, on my test, draw a little quick picture of the cell membrane because I could probably use that on a bunch of questions. So looking at this question, which one is something that I didn't mention? Well, we're going to kind of zone in on the nucleic acids because DNA, ATP, and RNA are not found in the membrane. All the rest are found there, and you would see this in your book in chapter, it's chapter 8. I'm trying to find the page for you. It's page 127 in your book. The thing that might have thrown you is E, steroids, because we kind of shorten the cholesterol when we talk about it to just say cholesterol. But just to clarify that the cholesterol we're talking about is called a steroid cholesterol. Okay. Number 41, the presence of cholesterol in the plasma membranes of some animals. So you're going to cover up all the answers and think about the point of the cholesterol, and that is on page 126, and it's at the bottom of the page, and it says the steroid cholesterol, which is wedged between the phospholipid molecules in the plasma membranes of animal cells, has different effects on membrane fluidity at different temperatures. At warm temperatures, the body temperature of humans, for example, cholesterol makes the membranes less fluid by restraining the movement of phospholipids. But because cholesterol also hinders the close packing of phospholipids, it lowers the temperature required for the membrane to solidify. Thus, cholesterol can be thought of as a temperature buffer for the membrane, resisting changes in membrane fluidity that can be caused by changes in temperature. So that is all about this steroid cholesterol. So the answer that has to do with temperature is A. Enables the membrane to stay fluid more easily when cell temperature drops. Okay, number 43. We had this question on one of our quizzes. What is one of the ways that the membranes of winter wheat are able to remain fluid when it is extremely cold? I'm sorry, that part of it got cut off. I can't resize the window once it's set. All right, and what was confusing to people was the difference between A and B, because we just talked about cholesterol being responsive to temperature, but that was when we were talking about animal cells, and here we're talking about plant cells. So the answer is A, that for a plant cell to remain, for the membrane to remain fluid, it's going to increase the unsaturated phospholipids in the membrane. So that's a key difference between plant and animal cell membranes. Number 45, which of the following is correct about integral membrane proteins? Now, this one would probably throw you because you might know what an integral membrane protein is and be able to picture that in your mind, but not understand its other term. So this comes from page 128 in the textbook, and it tells you right in the sentence that they can be transmembrane proteins, C. And I'm just giving you the answer here because at first this one threw me because I didn't think of interchanging those terms. So trans means across, and so we're talking about proteins that go fully across the width of the membrane. So an integral membrane protein can be one that goes across the width of the membrane. So the answer is C. Number 47, what membrane surface molecules are thought to be most important as cells recognize each other? So this comes from page 127, 128, and 129. The actual term is on page 129. It might be helpful to make yourself some flashcards of the different types of proteins found in the cell membrane because it's important to be able to distinguish them. So we know for sure it's not A because phospholipids are just making up most of the membrane. They don't have anything to do with how cells recognize each other. Integral proteins can be involved in transport. The peripheral proteins are the ones that are loosely bound to the surface. And the glycoproteins are the ones we're talking about here. And I'm trying to look for a good picture of them. But they're composed of a sugar and a carbohydrate. And if you remember what I demonstrated of how they work in class, I put my hand up and used it as a way to point at Josiah and say hi to him. And that was my way to recognize him. So that's what a glycoprotein is for. Okay, here's a straight definition type question. Which of the following statements is correct about diffusion? So you have to think about what diffusion is and remember that whether it's passive or active. And the trick that I remember diffusion by, which I gave in class, is that you spray perfume and the perfume molecules are going to diffuse from the bottle outward in a room, and it's not going to require any effort, and it's definitely going to go from an area of high concentration to lower concentration as it moves across the room. So the answer to this one is C, a passive process in which molecules move from a region of higher concentration to a region of lower concentration. It's certainly not rapid over long distances. It does not require energy. It's not an active process, and it doesn't need integral proteins in the cell membrane. Okay. 52. Water passes quickly through cell membranes because. So hopefully you're remembering how water passes through. The tricky thing here is that the bilayer is hydrophilic. Well, it has a hydrophilic portion, but also has a hydrophobic portion. So that's why water can't pass through the membrane without a protein. It moves through hydrophobic channels would never work because water is not going to be drawn somewhere that's hydrophobic. Water movement is tied to ATP hydrolysis. Nope. doesn't it doesn't pass quickly because it's a small polar charged molecule. That's why it can't pass through without a protein. And E, it moves through aquaporins in the membrane. And of course, that's the aquaporins. That's my trick. And that is indeed the answer. Okay, I know you can't read all of this one, so bear with me while I read it to you. A patient has had a serious accident and lost a lot of blood. In an attempt to replenish body fluids, distilled water equal to the volume of blood lost is transferred directly into one of his veins. What will be the most probable result of this transfusion? So, distilled water is being transferred directly into his veins. So, they want to know what the result will be on the red blood cells. So, this is a time to think about whether it would be hyper or hypotonic and what result that has on the cells. And I would always draw this out to make it easier. So if it's just water, of course, inside the cell is not just water. So where's the water going to go? Well, the water is going to move into the cell because the blood fluid will be hypotonic compared to the cell. So that's C. All right, and then looking at plant cells, celery stalks that are immersed in fresh water for several hours become stiff and hard. Similar stalks left in a salt solution become limp and soft. From this, we can deduce that the cells of celery stalks are, so when they're in water, they become hard, and when they're in salt solution, they become soft. So take a look at this. Is it hypotonic to both? Is it hypertonic to both? Hyper to the fresh water, but hypo to the salt. Hypo to the water, but hyper to the salt, or isotonic to water and hypotonic to the salt. And remember, of course, what plant cells' preferred condition is. Right, so the answer is C. They are hypertonic to fresh water, but hypotonic to the salt solution. Wait, does that make sense? I think the answer is D. I think this is a mistake. Oh, we're talking about the inside of the cells, not the solution. I get it. So it's C, because the cells are hypertonic to fresh water, but hypertonic to the salt solution. Hypotonic to the salt solution. If we're asking about the solutions, the fresh water is hypotonic to the cells. So in this case, they're asking for the cells. Ooh, tricky question. Good one. The membrane activity nearly opposite to exocytosis is, right? So some of the terms here might have been new to you. You probably had exocytosis before, but only relative to endocytosis. So which one of these is one of the endocytoses? Well, that would be D, phagocytosis. Remember the pinocytosis and phagocytosis. Cell eating, phagocytosis, and cell drinking, pinocytosis. All right, so moving on to cell signaling. Of the following, a receptor protein in a membrane that recognizes a chemical signal is most similar to take a moment to check those out. What are we making an analogy to here? So the answer here is A. And if you remember the idea that enzymes are kind of a lock and key situation and receptors and signals are kind of the same way. And I think in class Josiah actually brought this up, that he remembered that kind of locking in of the shape of an enzyme and its substrate. And that was similar to how a receptor protein is perfectly shaped for its signal that will attach to it. Right. Okay. Signal molecules. So this is one of the questions that I always hated as a student, where we had an A and B and A, B, and C. So most signal molecules, A, bind to specific sites on receptor proteins in a membrane. Well, yeah. B, are water soluble. Are they? C, are able to pass through the plasma membrane by active transport. Well, the answer here is D, A and B. So signal molecules are able to bind and water soluble. So this is one of those places where you probably knew A for sure, but we're not aware of the other qualities of a signal molecule. All right, so the water soluble quality of signals is on page 205 in your book, in case that's a little bit confusing. Okay, number 78, testosterone functions inside a cell by, and I'll give you a minute to read this. All right, the answer is B. So testosterone is a hormone. So it binds with a receptor protein that enters the nucleus and activates specific genes. So this is on page 205. In the picture, they show you the steroid hormone testosterone and its receptor, and how the hormone, so the hormone comes in, okay, it passes through the plasma membrane and binds to its receptor protein, and then the whole hormone and receptor together go into the nucleus of the cell through the pore in the membrane. Remember that we talked about the nuclear pores let particular molecules in and out, and the nuclear membrane allows the testosterone and its receptor to go in, and it says, hey, we're going to need to make a protein here. So a messenger RNA is made, and a message is conveyed so that a specific protein can be made at the ribosome. So the hormone testosterone is a signal for making a particular protein. Okay, I'm going to end part one of the review. I'm going to end part one of the review now because it's time for me to go eat my dinner, and I will continue with part two of the review later.